Today is National Bioenergy Day, so we are going to be presenting a webinar today. This comes as part of a series with the Advanced Hardwood Biofuels Northwest webinars. This is a project that is funded by the USDA, and we are working to make biofuels and biochemicals from hybrid poplar trees. The project has 6 universities and 2 industry partners across the Northwest working together to study everything from growing and harvesting the trees to converting them to fuels and chemicals. And today I am here at the University of Washington with Dr. Renata Bura and Dr. Ferando Resende, who are going to present on converting hybrid poplars to biofuels and biochemicals. We also have our project lead with us, Dr. Rick Gustafson, and he'll be able to join the discussion and answer any questions at the end. So a few housekeeping items before we begin. If you have a question, you can type it into the chat box on the left here. Everybody's microphone is muted. We can't hear you, so you'll have to type it in there and we will address it probably at the end of the presentations. So with that, I'm going to pass it over to Renata and Fernando.
Good morning, everybody. My name is Renata Bura, and it's a pleasure for me to present and to share our results with you with regard to conversion of lignocellulosic biomass, hybrid poplar, short rotation hybrid poplar, to be specific, to fuels and chemicals. Today I'm going to be presenting with Fernando and Rick, and is going to also join us for questions. We are here at the University of Washington, and all of us will work in biofuels and bioproducts lab here at the University of Washington. Right, so the objective of this talk is to share with you our biochemical conversion of short rotation hybrid poplar to fuels and chemicals. Probably some of you had a chance to see our previous webinars, which was actually presented by Greenwood Resources, our industrial partner in this research. And they really talk about how to grow hybrid poplar, what are the characteristics of the hybrid poplar, short rotation hybrid poplar. We're going to talk a little bit about the hybrid poplar today, but specifically we're going to concentrate on a bioconversion part. So what can we do? What kind of products we can get from this new feedstock? Right now, I want to share with you our hybrid poplar to fuels and chemicals platform. Okay. So in this one short snapshot, you're going to see what we do in our lab and how we actually can create these different cool fuels and chemicals from lignocellulosic biomass, such as hybrid poplar. So here I'm pointing right now at the hybrid poplar. You see a picture of the biomass. Short rotation hybrid poplar, you see lots of leaves, lots of branches. The stem is usually smaller compared to the mature trees, such as 8 to 12-year-old trees, and they have also lots of leaves. Okay, what's the chemical composition? I teach the whole class about chemical composition of lignocellulosic biomass, and I promise I'm not going to bore you with all the details. But what's the most important? Most important component of lignocellulosic biomass for us is our sugars. Our platform, poplar to fuels platform, is based on sugars. If we have lots of sugars in biomass, we're extremely happy. We love sugars. In lignocellulosic biomass, you have sugars, different types. They're big. At the same time, you have lignin, which glues all the sugars together and also glues extractives and other components of lignocellulosic biomass. In order to get into sugars, we have to use some kind of separation method. We have to basically get the sugars out of the biomass. I go to the gym maybe 3 or 4 times a week, but at the same time, if I get the handful of chips coming from hybrid poplar, I won't be able to squeeze the sugars out of it. So we need something stronger. Okay. So in our process, we use as a pretreatment, we use a steam pretreatment process. We apply steam to a biomass at high temperature and pressure, and therefore we can separate the main components, i.e. sugars, for example, from lignin and other components. Okay. And I'm going to talk about this steam explosion in details in a few minutes. So you see right now, you see a picture of our steam gun with Shannon and Rick. Okay, once we separate the components of our biomass, the sugars, again, we are about sugars in this process, they're too big to be used by microorganisms during the fermentation process. Okay, so it's the same like when you eat a steak. Okay, you have to use a fork and knife to eat the steak because the steak is huge. You can't eat it once. In our process, rather than using knife and fork, we use enzymes to break down these huge sugars, for example, cellulose, that's the name of the big polymeric sugars, into glucose. And once we have the smaller monomeric sugars, smaller components, smaller bites, the microorganisms, which we use in the fermentation process, which is next here, can actually utilize and produce cool products for us. At the end of hydrolysis, again, we break down the big polymeric sugars into small units, small sugars. At the same time, we have lignin. What do we do with lignin in our process? We can burn it to create energy for our process. All right, in the next step, so we have all the sugars in the right form. Our microorganisms are so happy and waiting for the sugars from us, and they can start fermentation. We can actually create different products during fermentation. If, for example, in a fermentation, we use 5 and 6 carbon sugars, i.e. variety of sugars, we can, and we use microorganisms such as Morella, thermoacetica, we can produce acetic acid, okay? If we use, for example, different microorganisms, for example, baker's yeast, we can produce ethanol, okay? Once we have, for example, acetic acid, I run across the room and I give my sample to Fernando's team, and they do the magic. When I say they do the magic, they do catalytic conversion. So this is the next step. So far, we talked about converting lignocellulosic biomass, the sugars from lignocellulosic biomass, and other components to, for example, acetic acid using biochemical platform. Right now, I'm going to talk a little bit, and Fernando is going to discuss in greater details the conversion of products, for example, acetic acid to jet fuel, for example, or other biofuels using catalytic conversion. So he uses chemical catalysts, not the microorganisms, but chemical catalysts and different types, and he's converting, like I said, the products coming from us to cool stuff. Okay. What kind of biomass we use in our process? All of you know at this point that we use hydropoplar, and hydropoplar we don't grow outside of our university. We get the hydropoplar from Greenwood Resources. And here you have a picture of hydropoplar trees. Again, they're 2-year-old trees, lots of leaves, branches, stem, big stem. Okay, and hydropoplar is grown by Greenwood Resources, and they have 4 plantations: Jefferson, Clarksburg, Pilchuck, and Boardman. And I'm giving you also some clones which we have and have been working or still working in our labs. So we work with different types of trees, hydropoplar trees in our labs. Right now, I'll just share with you briefly how do we get our biomass in terms of, okay, it's coming from Greenwood Resources, but how do we grow hydropoplar? So again, it's a very, very short introduction of how to grow hydropoplar. Hydropoplar is easy to cultivate and harvest. Okay, so what do we do? At year 0, we plant our trees. So basically, we can stick the stick inside of the ground and wait for 2 years, and it's gonna grow. And here you see beautiful pictures of hydropoplar. And after 2 years, we harvest. Okay, so you see a harvester right now, which is harvesting 2-year-old poplar trees. We harvest after 2 years because we want to establish the stump. Okay, and here you can see that there's a presence of stump after harvesting. Okay, and for us, it's extremely important, like I said, to establish the stump. So that's why we have to harvest after 2 years. The characteristics of the 2-year-old trees in the first cycle are totally different than characteristics of the 2 or 3-year-old trees coming from a second cycle, for example. Again, we are working with Greenwood Resources, and they're sharing information with us that the hybrid poplar trees from the second cycle, they have lots of big stems, less branches, and less leaves. So that's very important to us. So again, once we establish the stump, they reproduce, the hybrid poplar reproduces by couples, and we can harvest here you actually see the picture of the biomass after harvesting. So we can harvest after that every 3 years for 5-year cycle. Okay, so here you see trees in a second cycle. Okay, right now you know how we can grow the short rotation hybrid poplar. Let's talk about the samples which we have in our lab. We have a variety of samples coming from different sites. So in our lab, we worked in the past with poplar chips coming from mature trees, 8 or 12-year-old trees. We work with the whole tree mixture, and you've seen actually after harvesting, that's what we get. We get the tree mixture, lots of leaves, lots of branches, bark, and white wood. Also, we can manually remove the leaves, and in our process, we also in the past and currently using the poplar trees actually without leaves. We also converted in our lab just leaves by itself to sugars, and just to see how they actually, are they good for conversion or not. Okay, so we have different types of samples. Before I go actually into specific research findings, I want to share with you a little bit more information about how do we actually how our bioconversion platform works. So again, give you a little bit more details. I told you at the beginning, in our process we use steam explosion. Here you see actually in the middle the picture of the steam gun. It's actually inside of this shiny gray box. So what's a steam gun? When I say steam gun, what comes to your mind? It's not the big, gun, which, you've probably seen from Terminator or other Arnold Schwarzenegger movies. It's a just high pressure vessel. Okay, so we use our steam pretreatment process in a high pressure vessel, and we apply steam to our biomass, and steam comes from boiler. Here you can see on the left-hand side picture of the boiler, and everything is controlled and automated, and we use control module here. Great, so how the steam gun works? First, we have to insert our biomass into the steam gun. All right, so we open our fill valve, and you see right now biomass is ending up inside of the gun. Okay, after that we close our fill valve, we open our steam valve, and we steam our biomass for a certain period of time. Usually for hybrid poplar we use 5 minutes. Okay, and again the steam comes from the boiler. Then after 5 minutes we close our steam valve, we open our blow valve, and biomass is actually ending up in the receiving vessel. And here you can see a picture. After steam explosion, you can see a picture of the biomass, steam pretreated biomass, for example, hybrid poplar. It's a slurry, it's a mixture of solids and liquids. After separation, we can actually get the solid stream and liquid stream. So what's in the solid stream? Remember, I told you we are about the most important component of lignocellulosic biomass for us, it's actually sugars. So the name of the sugar which we have in the solid stream is called cellulose. We also have lignin, which glues, again, the sugars with the other components of lignocellulosic biomass. In the liquid stream, we have other types of sugars, acetic acid, sulfuro, and HMF. Again, what do we do with the solids? Remember, the sugars are too big. The same, I mentioned to you the steak and the knife and fork. So we have to use enzymes to break these big sugars into small monomeric units, such as glucose, for example. And glucose from the solid stream or from the liquid stream can be fermented to ethanol. Again, you can use baker's yeast, or it can be glucose and other sugars can be fermented by using morella, thermoacetica, and we can get acetic acid. And again, we can stop here, or we can be smart and clever, or I can just run across the hallway and give all the samples to Fernando, and Fernando can do magic with them. Fernando, now it's your turn.
Good morning. So this is Fernando Resende, and I'm going to be talking about the catalytic conversion and some of the products and that we obtain in the AHB process. So the first thing that I'd like to highlight here is the high flexibility that we have in this process in terms of products that we can actually make. There's a variety of fuels, but not only fuels, chemicals that are really important that we could be making as well. And the first example here is acetic acid. And acetic acid we obtain via fermentation, but it's a very important chemical. So that could be a product in itself, Acetic acid is present in paints and plastics, and it is a very important component of road de-icing material. So there is a market for acetic acid right now. So at this point, we could already make acetic acid via fermentation and use that as a valuable, high-value chemical. Now, once we have the acetic acid ready, there is also the option of combining that acetic acid with hydrogen molecules, as we see in the slide, and producing ethanol. So we're essentially hydrogenating the acetic acid molecule and producing ethanol, which we all know can be blended with gasoline. So that would be a case where we are producing fuel, and water is generated as a byproduct. Now, one important and natural question that could arise here is why would we go this route to produce ethanol? Why can't we just do straight fermentation to go to ethanol rather than trying to make acetic acid and from that acetic acid to make ethanol? And the reason for that is essentially related to the stoichiometry of fermentation. If you are fermenting, let's say, glucose to produce ethanol directly, you're gonna lose a substantial part of your carbon in the form of CO2. So your yields are gonna be limited by that stoichiometry. However, if you go through the acetic acid route, so you ferment to acetic acid and then you hydrogenate that acetic acid to generate ethanol, in theory, there is no carbon losses that would be anticipated there. So it would be, in theory, possible to convert 100% of the carbon of your fermenting material to ethanol. So there would be no losses anticipated there. Okay, then once we have ethanol made, we can use it as a fuel, but we can also dehydrate that ethanol and essentially eliminate a water molecule to generate ethylene. Okay, so that's another case of a different product that we can make with this process. And ethylene has a very important function. It's the largest commodity chemical in the world, and it's the raw material for a lot of the things we use in our daily lives, like plastic bottles and bags. So there is a huge market for ethylene that would be also readily available. So we have this option of switching between chemicals and fuels as this process gives us that flexibility. Okay. Now, once we have the ethylene, we could go and use that ethylene as raw material for other things, or we can still think about going all the way to jet fuels and diesel. And the process of converting that ethylene into jet fuel and diesel essentially takes two steps. And the first step is what we call oligomerization. So if you think of the ethylene as a single unit, oligomerization is just the process of putting a few units together and forming what we call an oligomer. So you're putting together maybe 4 or 5 units of ethylene and making carbon 10 or carbon 12, a relatively small molecule still, but larger than the original ethylene. Now, if you notice in the figure, as you do the oligomerization of ethylene, typically what you get is a higher olefin, which just means so you have carbon and hydrogen in that molecule, but you have a double bond that always stays at the edge, at the last carbon there. And if you want to go all the way to jet fuels, then you have to hydrogenate that bond. So that takes us to the last step, which is the hydrogenation of the oligomers. So you take the oligomers and add that to additional hydrogen to produce alkanes, which would be essentially the components of jet fuel and diesel.
All right, so now you know our all secrets, whatever we have secrets in our lab, how to convert short rotation hybrid poplar to, for example, biojet, not biodiesel, that's for sure. We want to share with you at this moment some of our highlights in terms of the results which we're getting from our labs. So specifically, I'm going to talk for a few minutes right now about what kind of products, what kind of sugar yield we get after converting 2-year-old hybrid poplar trees via steam pretreatment and enzymatic hydrolysis. Again, you've seen some pictures of 2-year-old trees and some biomass after harvesting. And you probably noticed that this biomass is extremely heterogeneous. When I say heterogeneous, you have lots of branches, bark, leaves, and whitewood. So I'll share with you also some physical and chemical characteristics of the biomass and tell you how we are dealing with this heterogeneous biomass in our lab. So in terms of experimental design, in our research, we use hybrid poplar trees, 2-year-old, coming from Jefferson, and we use three different types of mixtures. So mixture which was, this is the mixture of the biomass coming from 2-year-old, the whole tree mixture without leaves. Our undergraduate and graduate students worked for months to separate the leaves from the mixture. So we got the second sample, 2-year-old whole tree mixture without leaves. And also we wanted to convert leaves to sugars and see what kind of yields we're getting. Okay, all the experiments were performed in triplicates, and all the biomass was impregnated with SO2 prior to steam explosion. We used 3% SO2 concentration, and then we steam exploded our biomass using our great steam gun at 195 degrees Celsius and 5 minutes reaction time. Okay, what are the physical characteristics of our biomass? You've seen it before. It's extremely heterogeneous. So here in the middle, in the pie graph, you see the physical characteristics of the 2-year-old poplar mixture. It was composed of chips, 42%, this is violet color, leaves, 37%, this is a blue color, branches, 12%, red color, and bark, green color, 9%. By removing the leaves, we can increase the chip content from 42 to 67%, right now on the other graph on the left-hand side. We can also increase our branch content from 12 to 18 and our bark content from 9 to 15%. Again, the third series of samples, we use just the leaves which were separated from the mixture. And here actually you see the pictures of each of the components, leaves, bark, branches, and chips. You see they look different, and they have also different chemical characteristics, as you see in a few seconds. On this table, you see chemical composition of 2-year-old hybrid poplar trees, each of the fractions. In the first column, you see leaves, branches, bark, and chips. And then in the first row, you see sugars, arabinan, galactan, glucan, and xylan, and mannan. Total sugars, we added all the sugars together. Total phenolics, ash, and extractives. And I highlighted for you 2 rows, leaves and chips in yellow. Okay, now let's take a deeper look and see the chemical composition of leaves. They don't have much sugars. The total sugar content is 21%. They have lots of phenolics, around 40%. They have lots of ash, and they have lots of extractives. Again, at the beginning, I was telling you, our process is based on a sugar platform. The more sugars we have in biomass, the more happy we are, right? We do the happy dance if we have lots of sugars in our lab, right? So you see, leaves are not so good really in terms of the sugar content. They don't have enough sugars for us to be processed. But let's compare with chips. You see they have almost 30% more sugars. They have less lignin, and lignin, again, we can burn in our process the lignin to create energy, but again, lignin is the glue. Remember, the more lignin we have, then we have to sweat more in our lab, and we don't like to sweat in our lab a lot, right? So, and then they have less ash. Ash is not so good for the process, and also they have less extractives. So chips, obviously, they're way better for converting to sugars and creating these super products than leaves. And again, in terms of chemical composition of branches and bark, there's someone in the middle. All right, so what about the chemical composition of all the three mixtures which we use in our lab for this set of experiments? Again, in the first column, I'm showing you a 2-year-old poplar mixture without leaves, chemical composition of the 2-year-old poplar mixture, including the leaves and just leaves by itself. Again, here in the first row, you see a concentration of all the sugars, total phenolics, ash, acetic acid, and extractives. And again, I highlight for you the first 3 rows. I mean, actually 2 in yellow. Okay, let's take a look at total sugar content. Okay, by actually removing the leaves from the mixture, we can increase the sugar content from 41% to 50%. So that's good. We love sugars. We can also decrease our lignin content by removing leaves from 28% to 25%. We can also decrease our ash content from 5.7 to 3%. We can also decrease our extractive content from 15 to 12.7% by actually removing leaves. So that's fantastic. And again, you've seen before, leaves are not so good for a conversion because they don't have much sugars to begin with. All right. Now you know what's the physical and chemical characteristics of our biomass. Let's discuss how do they behave during the pretreatment and hydrolysis. So we've done all the pretreatment. We used all the forks and knives to cut these big sugars, cellulose into small monomeric units. And here I'm showing you how much sugars did we recover. These monomeric sugars we recover after pretreatment enzymatic hydrolysis in kilograms per ton. From the mixture without leaves, from the whole mixture, and from leaves by itself. And on y-axis here, you can see kilograms of sugars in monomeric form, which are ready, for example, for fermentation. And after fermentation, like I said, we can just go and give the samples either of acetic acid or of ethanol to Ferando. So kilograms of sugars per ton of biomass. Again, you can see by removing the leaves from the mixture, we can increase our sugar content by 50%. Okay, so that's fantastic. We can go from 170 kilograms to 350 kilograms per ton of biomass. So that's really, really good. Leaves are problematic for us. They have low sugar content, and by removing the leaves, actually, we can increase our sugar yield after pretreatment hydrolysis. So that's fantastic. At the same time, as you can see from the last bar, leaves by itself, again, they didn't have much sugars to begin with, and they're difficult to hydrolyze and to convert. So they're not really good material for bioconversion, especially if you use a sugar platform. In conclusion, we can see that the 2-year-old poplar mixtures were composed of chips, leaves, branches, and bark. Again, very heterogeneous mixture. By removing the leaves, we can increase the sugar content by 11%, decrease lignin, ash, and extractive content. And also, by removing the leaves during bioconversion, we increase the sugar content after pretreatment and hydrolysis by 50%. So that's fantastic. At the same time, what we're learning, we're learning more about this new feedstock, the short rotation hybrid poplar. The 2-year-old mixture is very heterogeneous biomass, and again, we're getting more and more information how to handle doing the conversion process. You see on the left, the white, good-looking chips. We know what to do. We've been working with them for past, 15 years because they're coming from mature trees. But right now, we're learning and researching how to deal with this new mixture of the short rotation hybrid poplar. And again, we are so excited at this time because right now, Greenwood Resources just harvest for us just a few trees coming from the second cycle, and they're fantastic. They have huge stems. We love stems. Remember, we love lots of sugars, and the sugars are usually in the chips, so in the stems. They have less leaves. We're not crazy about leaves, but we understand that the trees, they have to have leaves. We know that. At the same time, they have less branches. So the new crops coming from the second cycle, it's way better in terms of productivity, and it's way better in terms of chemistry and processing, and we are so excited to work with them right now. Would you like to do questions?
Okay, so in the sequence here, assuming we have the ethanol as a product from the previous steps, and now I'm going to be talking about some of the results that we have been able to, uh, some of the things we have been able to accomplish by converting ethanol into jet fuels and diesel for precursors of jet fuel and diesel. So let's go over the steps again. We have three major steps to convert alcohol to jet. And the first one, we start with ethanol, dehydrate the ethanol to make ethylene. Water is left as a byproduct. Then we go through the second step, which is the oligomerization of ethylene, where the ethylene, which is a single unit, a few units are put together in a heterogeneous catalytic process to produce the oligomers. And this step, the oligomerization of ethylene, is really where the major difficulty is, and that's where most of the work that we have done has focused, because there's a lots of difficulties associated with this specific step. Then once you have the oligomers ready, again you have the double bond at the end of the chain, so you use hydrogen to hydrogenate those, and then you produce jet fuels and diesel molecules, alkanes, that way. Okay, so a process diagram here, just so that we can go over how this works. The ethanol is fed to a dehydration reactor. We see here a separator where we get the water out, and then the ethylene is fed to an oligomerization reactor. And then out of the oligomerization reactor, you get your product, but part of that product, the shorter chains, hydrocarbons C4 to C8, they can be potentially recycled if you are producing jet fuel and diesel, because you are really aiming the longer chains here. Now, the longer chains, once they are made, C10 plus olefins, remember we still have olefins, they can be fed to the hydrogenation reactor with hydrogen to then produce jet fuel and diesel molecules, okay? Now here's some of our previous work that has been done with Ziachem and on the dehydration of alcohol. So this step, we completed the experiments using a commercial alumina catalyst. So what I really would like to highlight here, the dehydration step is really quick and straightforward. So the catalyst that we have used is inexpensive, is readily available. As you can see in the plot, conversion as function of the weight hourly space velocity here, it is possible to keep the conversion and also the selectivity to ethylene higher than 99.5%. Now, in addition to doing the work with ethylene, we also have been able to do experiments with propanol, so the C3 platform, and then that platform, we are essentially using the same catalyst and obtaining similar results. So C2 and C3 were both performed. On the right, you see a picture of the fresh catalyst and the catalyst after 310 hours. There's not really a whole lot of sign for deactivation there, just a change in the color of the catalyst. Now let's focus on the oligomerization of ethylene. So in this step, you have the unit ethylene being so several units being put together to form the oligomers. One of the characteristics of this specific reaction, it is exothermic. So it is difficult to control the temperature in the system. It releases heat, and if the temperature starts going too high, the process becomes very difficult to control. And in that case, we have a secondary reaction happening, which is the formation of aromatics. So aromatization of ethylene is also a possibility when we are working with these systems. However, that's not the type of molecule that we are trying to produce here, aromatics. The combustion of aromatics is typically unclean. It generates significant amounts of smoke and char. If you have benzene, for instance, in gasoline, so benzene is known to be a carcinogenic compound in gasoline, its limit is only 0.8% in weight. And if you're talking about the entire set of aromatics, the limit is 20% in gasoline. So there's a limit of aromatics that you want to produce. And in our case, we really want to avoid that route, not only because of the unclean combustion of aromatics, but also because they can recombine and forming larger structures, forming polyaromatic hydrocarbons or coke, which can stay inside the catalyst pores, preventing the reaction from happening further, essentially deactivating the catalyst. So the route that goes from ethylene to aromatics via aromatization and then formation of coke, that's really what we want to avoid. We want to just stay with the top reaction where the ethylene combines several units to produce the oligomers. So the work that we've been doing in our lab has as major goal to produce lab scale samples of diesel and jet fuels. And within this goal, we identify three main tasks. One of them is to tune the product selectivity to minimize aromatics. So again, we want to produce as little aromatics as possible and just focus on the oligomers. As we are minimizing the aromatics, we are also hoping to minimize the coking, which is associated with the aromatics formation. And along with that, we are also investigating the effect on the catalyst support. So here we see the experimental setup that we have. It's a packed bed reactor. It's essentially an empty tube where we place the catalyst and the catalyst in a bed. Ethylene and nitrogen both flow through the reactor. What we have seen is that the nitrogen it's really an important, it is a carrier gas, has function of carrier gas here, and it's really important to carry the oligomers out of the system once they are formed. If we do not have the nitrogen, the tendency is that the oligomers just stay in the bed and they just keep reacting and forming coke. Okay, once the products are formed, we flow them with any residual ethylene through a condenser unit, a heat exchanger that cools down the temperature of the vapors to about 5 degrees C. And once that happens, then you condense your products. The oligomers are obtained in liquid form, and any gas that is left after that flows through a vent, and we collect a sample for GC analysis with an FID and a TCD. This gas contains any residual ethylene from the oligomerization step, so that's how we actually calculate the conversion of the process. Now, this is the strategy that we use to convert the ethylene into oligomers. We used bifunctional catalysts in our packed bed reactor. So essentially what this is, as the figure shows, we have a support, which is typically a zeolite material, and this material has a catalytic function, while the metal itself, which in the case of oligomerization is typically nickel, so the metal it's actually the most active species. So both the metal and the support have a function in the catalytic reaction, so we use this bifunctional catalyst in the process. That's the strategy that we use for the oligomerization process. We have been able to achieve really good results on the oligomerization front. We had a breakthrough where we were able to synthesize our own bifunctional catalysts, and we had a couple of them that we were able to successfully produce and use in the oligomerization system to produce liquid oligomers. And here you have a picture of what a liquid sample looks like, some of the major molecules that were obtained. This has been done in the literature work before the oligomerization of ethylene. However, most of the previous work is done in microreactors, so you don't usually see samples like that as the ones that we collect. Okay. One of the keys for the success of this bifunctional catalyst is really the effect of porcine. So if you try to use as the support a more common zeolite material like HDSM5, for instance, the pores of HDSM5, they're extremely small. They're about the same size as a benzenic ring. So once you have that reaction happening in a support like HDSM5, the tendency will be hydrocarbon molecules will be accumulating inside those pores and just growing, reacting further and further, and that just leads to coke formation and deactivation. However, the supports we used in this work, and I'm going to mention here aluminum MCM41 and H-beta, these are mesoporous supports rather than microporous, and in these larger pores, they allow larger molecules to move in and out of those pores in an easier fashion, which means you have less problems with coke formation and deactivation that way. Now here we are showing some of the results that we actually obtained with some of our catalysts. So nickel aluminum MCM41 was one of the catalysts we tested, and here you see the catalyst preparation and the sample. The carbon number that was obtained in this out of this experiment, we go from C4 to C8. So we are more in the short chain size oligomerization with the specific catalyst. But at the bottom here, we see when we compare the types of products that were made, we essentially had almost 100% olefins. So we really were able to focus on the oligomerization reaction. There were a little bit of paraffins and also naphthens, but essentially no aromatics were produced. So we achieved the goal here of eliminating the aromatization reaction with this specific catalyst. Another catalyst that we have used as well, and that was also a successful catalyst, was nickel H-beta. And here again you have the catalyst preparation and the liquid product. H-beta had a much wider product distribution. So with this catalyst in a single step, we were able to produce hydrocarbons all the way from C4 to C16. And when you look at the types of products that you made, still the majority of it was olefins, while we had some paraffins and naphthens, a very minor amount of aromatics. So with this catalyst, we're also able to minimize the amount of aromatics produced. Now, the difference between these two catalysts that I have shown, H-beta and MCM41, is essentially the acidity of each catalyst. So we see nickel MCM41 produced a narrower product distribution as far as the carbon length, the chain of the length, the length of the chain, and then nickel H-beta went all the way from C4 to C16, and our hypothesis here is that the higher acidity of the second catalyst, H-beta, this catalyst has more, the support has more aluminum, it is more acidic, so we have hypothesized that this higher acidity has contributed to further oligomerization reactions and therefore the higher order of oligomerization we have observed. So as a summary here, we did the alcohol to hydrocarbon process, and we had a breakthrough on the oligomerization of ethylene with two catalysts, two bifunctional catalysts, nickel MCM41 and nickel H-beta. In both cases, olefins are the primary product. We have either no aromatics or almost negligible aromatics obtained and collected in the liquid product. And for the next year, what we are planning moving forward with this process is to work with Giachem and the scale up to gallons. So that's essentially the idea. A lot of the literature work has been done with microliter. In the microliter scale, microreactors, we have been able in the AHB project to do milliliter scale to generate laboratory size samples of the oligomers, and then moving forward, we're going to be working to expand that and scale up to gallon scale, working with Giachem. Now, let me give you a more general summary of the project, of the results that we talked about here. Short rotation hybrid poplar, physically and chemically heterogeneous biomass, a very heterogeneous type of material. You saw Renata talking about the leaves. But we were able to produce successfully sugars from short rotation hybrid poplar, and in addition to that, we were able to successfully produce jet fuel precursors, the oligomers, starting from ethanol. So here I would like to just make a stop and acknowledge all the people that worked in this project. We have graduate students, we have undergraduate students, we have postdocs. There's a number of people working both in the biochemical conversion and the catalytic conversion. So these are really the ones that have been able to move this project forward and generate the results that you see in this work. And I also would like to acknowledge the USDA National Institute of Food and Agriculture for the grant that made this work possible.
Well, thank you, Renata and Fernando. It looks like we have some questions already over here, so I will read those off and they can address them. If you have more questions, please type them into the chat box. So the first one is, how sensitive is the process towards the moisture content of the wood? I think this is referring to the steam explosion process.
So basically, during our research with other types of feedstock, we use different types of wood and moisture content. If you have a biomass with a moisture content of 50%, it's really good, i.e. You can dry the biomass and you can re-wet it, but because we use SO2, and SO2 for us, it's if you have a biomass which is extremely dry, i.e. like maybe 2 or 3% moisture content or up to 5%, we're going to have a harder time to solubilize hemicellulose because you need some liquid to solubilize hemicellulose during steam explosion. So once you have above 30% moisture content, it's no problem. Like I said, when we work with just a hybrid poplar, not short rotation hybrid poplar, but the mature trees, the chips, once, like I said, you are above 30%, there's no problem. But we can always re-wet it. When I say re-wet it, we can just pre-steam our biomass prior to steam explosion, and we get the same results as for never dried biomass. For this research, actually, for this project, we always use biomass which was freshly cut, and then we freeze our biomass. The reason why we freeze our biomass because we want to have a continuation of the samples. But like I said, we've done for other type of poplar, the mature poplar, a study when we look at the influence of the moisture content. And like I said, you can dry it and re-wet it, or you can just have a fresh biomass. But it's good if you have a biomass when we use SO2 with the moisture content higher than 30%.
Great. And then Jeff asks, with the steam explosion, how many kilograms of fermentable extract is produced per kilogram of dry chips?
Okay, so when you have a dried chips, like I said, I assume if we were dried chips, it would be tough. Like I said, we have to have a certain moisture content. We can re-wet it, but let's assume our chips are 30%. So when we talk about how we compare our results coming from 2-year-old trees to mature trees, obviously they have a higher sugar content to begin with. They have 8 to 12 years to grow. Our trees, especially, again, the results which I was showing, they're coming from first rotation. So after 2 years, they have to establish the roots and stamp and stuff like that. They, on average, 10 to 20%, they have a lower sugar content compared to mature trees, again, because they have a shorter cycle. They grow only for 2 years instead of 8 or 12 years. However, let's assume, and the conversion, it's a little bit lower when you compare white chips with 2-year-old hybrid poplar trees, but on average, when I was showing you the last graph, monomeric sugar recovery after pretreatment hydrolysis, we're going to have around 20 to 30% lower sugar content. But again, it depends on what kind of clones we're using and what kind of what kind of material we're comparing with. It is 8 or 12-year-old. However, like I said, we're excited about the new second rotation crop, and they have higher sugar content because they have bigger stems and they have less leaves and they have less branches. So they really, and they're extremely productive. So when you compare to the second rotation, for example, 2-year-old or 3-year-old coming from second rotation, they have way higher sugar content. And we are doing this analysis right now. So probably in 6 months or 3 to 6 months, I'll be able to tell more about the trees coming from second rotation. Like I said, first one, we're expecting to have lower sugar content because they have to establish all the root system. They have to establish the stem. There is sprout from the stem, so that's important. So they're not as productive as these trees from the second, third, and fourth rotation.
And finally, how common are bifunctional catalysts?
They are very common, and it's a common practice to combine metal catalysts and supports like zeolites, mineral materials for many types of processes. Even for the oligomerization of ethylene, the use of nickel on supports and inert materials, zeolites, it's relatively common, yes.
Well, if there are any other questions, feel free to type them in. If not, please fill out the poll questions on the right here. And thank you all for attending. And thank you to Renata and Fernando for your participation. Our next webinar that we're going to have is in January. We don't have a date yet, but you can check our website or our emails for a webinar coming up on regional economic impacts of a poplar-based biofuel and biochemical system. So thank you.